The complete DNA molecule consists of two antiparallel strands. Because the glycolic bonds between the sugar and base are not directly opposite each other, the molecule twists into a right-hand helix with two distinct grooves. The major groove is wider, deeper and more accessible to proteins, showing a more distinctive pattern of hydrogen bond donors/acceptors and methyl groups for each base pair. It allows for sequence-specific recognition by proteins. The minor groove is narrower and less accessible, and the base pair edges are less chemically distinctive. The major groove plays a role in protein to DNA binding. Transcription factors, polymerases, and repressors are able to recognize specific sequences within it. Additionally, it plays a role in DNA methylation recognition. Both the major and minor grooves are involved in gene regulation, providing binding sites for regulatory proteins such as transcription factors, enzymes, and some drugs. These proteins use the chemical information expressed and exposed by the grooves to recognize specific DNA sequences and regulate transcription. Some anti-cancer and antibiotic drugs bind in the minor groove, such as netrospin, which binds in AGT-rich regions, altering DNA conformation and blocking transcription factors. The two strands comprising DNA run in opposite directions. Their orientation is antiparallel. One strand runs in the 5' to 3' direction. This is the leading strand. The other strand runs in the 3' to 5' direction. This is the lagging strand. Antiparallel alignment allows the nitrogenous bases to face each other correctly and form hydrogen bonds at the correct angles. If both strands were to run parallel, the geometry would not allow stable hydrogen bonding, as the helix would not form correctly. Antiparallel orientation additionally has relevance to DNA replication. DNA polymerases can only add nucleotides at the 3' hydroxyl group end of a growing strand. Because the strands are antiparallel, DNA replication is semi-discontinuous. The leading strand is synthesized continuously in the 5' to 3' direction, while the lagging strand is synthesized discontinuously as Okazaki fragments, also in the 5' to 3' direction.